It's going live now. I like to start it right at 3:59ish. This way, less editing.
Yeah, exactly. How you doing, Dr. Parney?
Very good, thanks. Thanks for the opportunity to do this.
No, thanks for coming along. Get going in about 2 minutes.
Good afternoon,
Hey,
everyone.
Hey, Dr. Sheehan, how are you?
I'm doing well. Thanks for joining us, Dr. Gilbert.
All right, we're giving it one more minute and then we'll get going.
My pleasure. Thank you for inviting me.
Who wants to lead off? Dr. Parney, Dr. Gilbert, from your side of things?
I mean, I'm really here just as one of the editors of the special edition. I'm happy to give a brief introduction. Is Dr. Fetchy is going to be joining us, or?
I believe so.
Okay.
He had responded, yeah.
Yeah.
Ian, thanks for joining too. I didn't see you on the.
Oh yeah.
I
No,
just
my
scroll
pleasure.
over a bit more.
Yeah.
Well, it's 4 o'clock now. Why don't we get started and we'll let hopefully Dr. Fetchy is able to come along as well. Welcome everyone. Welcome back to the 2021 version of Tumor Talk, which is pretty similar to the 2021, but I think we're all in better spirits about it, hopefully. We'll see. But anyway, as everyone knows, this is a joint effort between the Journal of Neuro-Oncology and Lenox Hill Neurosurgery, where we like to focus on recent publications in the Journal of Neuro-Oncology, talk about the reasoning, the rationale, the results, and how it affects neuro-oncology and surgical neuro-oncology as well. And so today, delighted to have two special guests, guest editors of a new special edition of the journal on immunotherapy. And so Dr. Parney, Dr. Gilbert, if you want to take a moment and introduce yourselves, and Dr. Parney, talk a little bit about the journal and how, or the issue and how it got started, feel free.
Sure. So I'm Ian Parney. I'm a neurosurgeon at Mayo Clinic in Minnesota, and actually Dr. Miriam Rahman and I were fortunate to be the guest editors for a special edition on brain tumor immunotherapy in the Journal of Neuro-Oncology. Dr. Rahman is delayed a little bit. I know she had a case, emergency case in the operating room. Hopefully she'll be able to join us in a little bit. The special edition covers a wide variety of topics of tumor-mediated immunosuppression and immunotherapy in brain cancer. And we've been fortunate to have Dr. Gilbert, and we hope Dr. Fetchy will be joining us shortly to present a little bit about some of their papers, which we felt were particularly compelling. And we'll maybe turn it over then to Dr. Gilbert, who we're very fortunate to have join us here today as well. Excuse me.
Great. Well, thanks, Dr. Parney, and thank you all. A great pleasure, honor to be part of this program. I've seen some of the prior discussions, and I think that it's a terrific format, and I really appreciate that it's both low-key, laid back, and controlled in time. So I think a great opportunity to have important discussions about things that impact patients with brain tumors. For myself, I'm a neuro-oncologist. I'm currently a senior investigator and the chief of the neuro-oncology branch at the NIH. And I lead our translational immunology program. And what I'm talking about today are the effects of corticosteroids, particularly on T-cell mediated immune response. And this is in reference to a review paper that I wrote with one of our former fellows, William Kelly, who's now a neuro-oncologist at UT San Antonio. And So I think as everybody who is attending this conference knows that we've not done extremely well using immunotherapy for primary brain tumors. And this is in contradistinction to how well they're doing for some metastatic disease, brain metastasis with melanoma, where they're seeing upwards of a 50% response. However, in the context of brain tumors, the pediatric patients with the mismatch repair deficiency glioma, we are seeing prolonged responses. So there is the possibility of not only getting an immune response and navigating all of the issues like cell trafficking, et cetera, that I list there with immunologically cold tumors, et cetera. But of course, the low mutation rate of gliomas is circumvented with the mismatch repair disease. So there is hope. But I think we really underappreciate the fact that a lot of the things that we do to our patients does suppress immune response, in addition to all the issues that come inherently with the cancer. And here I point out the lymphopenia, which is very common with both radiation therapy and temozolomide, which is our #1 chemotherapy. And of course, what I'm going to be talking about is corticosteroid use. And glucocorticoids, as you can see in this little diagram, have a tremendous amount of effect on immune cells. Certainly don't have the time to go through all of this. This is a really nice review from 2011. And actually in the paper that Dr. Kelly and I wrote for Journal of Neuro-Oncology, we've also articulated the effects. And there's obviously a wide spectrum of effects on the various components of the immune system. You can see the T cells, the CD8, CD4, the B cells, myeloid cells, et cetera. So there is a lot of impact of corticosteroids on immune function. Looking at T cells in particular, I think is really important. And again, another diagram, another figure from our paper looking at the T cell signaling. And here you can see the molecular pathways involved with the antigen presenting cell, targeting the T cell receptor, and all the downstream effects that help the T cells actually kill cancer and do their function. But corticosteroids inhibit the T cell receptor signaling, decrease the phosphorylation downstream, so impacts on the T cell receptor CD4, CD3 interaction, and actually can trigger immune cell apoptosis. So that would decrease the lymphocytes. And so do corticosteroids affect immunotherapy for intracranial tumors? And so here, early on, there was a lot of disconnect in the sense of corticosteroids and their potential harm in the use of immune checkpoint inhibitor. And part of it is, as I will show you, was really a misunderstanding. So we use corticosteroids for organ transplant. We give it then before the transplant to suppress the immune response. In cancer immunotherapy for systemic cancers, they only give the corticosteroids after the patient has an immune toxicity, which by definition means that they've had an immune response. And then they point out, of course, that patients who require corticosteroids for immune toxicity do as well as patients who don't require corticosteroids. And so when you say, well, we think corticosteroids are impacting immune response in patients with brain tumors, they're saying, well, what do you mean? It doesn't seem to affect the patients who get it for systemic cancer. And of course, our patients are often on corticosteroids as part of their brain tumor management. So they start their immunotherapy often on corticosteroids, which is different than starting it after. So what is the proof of that? And I'd like to show you some data from work done in my laboratory trying to address this question. And this is a paper that was published a couple of years ago in the Journal of Immunotherapy of Cancer. Amber Giles, who was a postdoc in my group, did this work. And what she did was took donor T cells, so normal T cells, and asked the question, what do corticosteroids do? And you can see it has a pretty profound effect on proliferation, expansion, and precursor frequency. This is just a flow to show that the proliferative rate goes down and that they get stuck in G0, G1. So there's actually cell cycle arrest with corticosteroids, and proliferation goes down as shown up in the left-hand corner. And importantly, the naive T cells, that's Tn in this chart on the right, you can see that their proliferation of cell numbers go down, much less effect on the more mature T cells. So maturation does matter. And what we also found is the dexamethasone increased the immunosuppressive molecule CTLA-4 on the T cells. So that's a secondary effect. And again, in tumor-bearing animals with steroid exposure, we also saw CTLA-4 expression increasing, which was dose-dependent on the dexamethasone. And fortunately, there was some reversal when we used a CTLA-4 blocking agent, in this case, a CTLA-4 antibody. So when we did this in vivo in a syngeneic mouse model shown here, what we found is if you gave dexamethasone 7 days after a CTLA-4 blockade, you did see a therapeutic benefit to the immunotherapy. And so we felt that these animals actually didn't have the same situation. They did not have exhausted immune system because these were new cancers. And we also proved that actually if you just add dexamethasone a day after exposure in vitro to a stimulation, that you didn't block it. So you had to have the dexamethasone there at the time or before. So timing does matter. When we did the same in vivo experiment, now starting the dexamethasone a day before a tumor implantation and checkpoint inhibitor, we lost the beneficial effect of the immune checkpoint inhibitor. So I think important implications for clinical research. And there's been a recent paper actually published just a few weeks ago from our colleagues in Boston. David Reardon led these efforts, which again confirmed when they gave corticosteroids before anti-PD-1 therapy, it also abrogated a lot of the effect in their syngeneic in vivo model. So I think in summary, we've not seen a lot of success in primary brain tumors. There's been a lot of reasons that have been postulated, a hostile microenvironment. But one of the things we can control are the iatrogenic causes like lymphopenia and corticosteroid use. Our data suggests that the corticosteroid use blunts the immune response, particularly the naive T cells, which are actually you're counting on to be the ones to react to the new antigens from the cancer, increases the CTLA-4 expression, and hopefully future clinical trials should limit or eliminate corticosteroid use. However, the question that still remains out there is how long after corticosteroids are stopped is this inhibitory effect? How long does it remain? So I'm almost certain that a couple of days after you stop steroids, you will still have a blunted immune response. We just don't know how long it takes to reverse it. So that's just some background, and hopefully we can have conversation about implications of this work and how we can hopefully do better clinical trials in the future. So I will end there.
Thanks so much. I think, very provocative. This is obviously something that we all do. Kind of, it's the first thing you do, right? Is throw steroids at every tumor that comes in. If there's even a hint of edema, as long as you're not concerned about a lymphoma. Do you think? Number one, has this influenced your own practice? Are you actually recommending not to start steroids, to hold?
Well, so we're actually in all of our immune therapy clinical trials, we're not allowing patients to be on corticosteroids, and we're actually making it that they could not have been on them for a week. Now, where does a week come from?
Yeah.
I was going to say it's a round number, but it's not. It's 7 days. It's even a prime number, but a week sounds reasonable. We actually do have a clinical trial, which we launched pre-COVID, which we had to stop during COVID, where we are enrolling patients who we are tapering off of corticosteroids and monitoring their T cell function. We have an assay that we've developed in the lab, which gives us a sense of their activatability. It's an assay we developed and published in that paper in 2018. So we are trying to answer the question, how soon after a patient's been tapered off of corticosteroids do we see a reactivation of the T cells in response to a checkpoint inhibitor?
Mm-hmm. And what do you think about post-operatively? I mean, let's say you obviously, it depends on the treatment, I guess, right? It depends on the immunotherapy. But let's say it's an immunotherapy based on the tumor tissue itself, something like the old heat shock protein or the dendritic cells. Would we typically discharge a patient on a standing dose of steroids after their surgical intervention. Now, at this point, you're not getting rid of, you're not doing the pre-treatment really steroids to some degree. I guess you are because it's post-operatively. But I'm just curious what you think. Is there any role for them, or would you like to see them kind of phase out completely and just stop like a week or 2 weeks once you know better what the timing is?
Well, so, I think that there are lots of complication side effects from corticosteroids. So if we found, an alternative, that would be fantastic. I don't know what the implications are for things like heat shock protein, like other vaccines. I would be very curious to know what the tumor microenvironment would look like in a newly diagnosed glioblastoma where the patient has not received preoperative steroids. We just don't have that information. And I do recognize that it is absolutely the standard of care. I've done an unofficial survey of colleagues, neurosurgeons. And, of course, the concern is this theoretical issue about corticosteroids versus what if there is a problem with perioperative edema, right? You're in there and the brain hasn't been, quote, cooled down with steroids, all to hopefully maintain a microenvironment that you don't even know exists. So if there was an alternative mechanism by which edema could be controlled and samples obtained, and then we could do a comparison, again, a statistical comparison from a group of patients who've not had corticosteroids in the preoperative period versus those who have. And if we do see an alteration or an improvement in the microenvironment, would that be an impetus to find alternative mechanisms, particularly in patients who had interest or we felt would maybe potentially good candidates for immunotherapy? Are we compromising our immunotherapy potential right out of the starting blocks with the initial operation? And I don't know the answer to it.
Yeah, I mean, it's definitely tough to study without a doubt, and but a very interesting, provocative question. So it looks like Dr. Fetchy's not here yet, but Dr. Parney, we kind of alluded to this. If you want to just give some more highlights of what the journal, what the special edition has involved with it, we can at least let people know to check it out. And
right. So the special
points.
edition, and hopefully Dr. Fitch will be able to join us here in a little bit. I can go through a little bit of what he was going to talk about. I have to be without the 8 slides, but the special edition really covers a lot of different aspects of tumor-mediated immunotherapy or tumor-mediated immunosuppression and tumor immunotherapy for brain tumors. And so that covers the gamut. We've heard some of the things Dr. Gilbert just spoke about in terms of the effects of corticosteroids and really getting down to the nuts and bolts of what that's doing, rather than sort of a gestalt that we know that steroids are immunosuppressive. Dr. Fetchy and others have sort of talked about other aspects of systemic immunosuppression and local immunosuppression within the tumor microenvironment. And then there's discussions of different types of immunotherapy approaches, checkpoint inhibitors, vaccines of varying descriptions, dendritic cell vaccines, other vaccines, and combinations of these therapies, along with sort of the general idea that's becoming more and more apparent that combinations of these therapies are going to be really important or have the potential to be really important going forward. There may be some synergy between things that we're doing with surgery, radiation, chemotherapy, standard chemotherapy, and some of the immunotherapies that are being brought on board. This, of course, has implications for what we were just talking about. We might say, well, we'd want to have a combination of surgery and an immunotherapy, but that brings up the issue of steroids. And I'd say, well, maybe we'd want to think about using something other than steroids, and so the best default that we have at the moment might be bevacizumab. But then that brings up the issue of how are you going to combine that with surgery and so forth with this. So these are complicated issues that we really have to work through. I thought what I'd maybe just talk about a little bit, and then we can open this up for discussion as well, is some of the systemic immunosuppression that we see in glioblastoma patients, even above and beyond the corticosteroids. So dexamethasone is immunosuppressive, and as we heard about that, that's critically important. And we actually know that patients with glioblastoma who are on dexamethasone can have CD4 counts as low as patients with full-blown AIDS. They may not be as functionally immunosuppressed, but they're definitely immunosuppressed. But even in the absence of corticosteroids, glioblastoma patients actually can be pretty profoundly immunosuppressed systemically, which is kind of interesting. And when you think of, typically you think about a glioblastoma, you think, well, this is a unisystem disease. It just involves the brain or the central nervous system. But in fact, it can have pretty profound systemic effects. And, some of these are things that we've been aware of for a while. We know that the tumors secrete cytokines and other things that can be immunosuppressive. That seems to be more an issue of something that's happening in the tumor microenvironment alone. For some of the things that we know that are secreted by glioblastomas that are immunosuppressive or at least immunomodulatory, like TGF-beta or IL-6, or at least from within the tumor microenvironment, maybe not from the GBM cells themselves, IL-10. They're elevated within the tumor, but they're not necessarily increased systemically. So there may be something else going on. And I think Dr. Fetchy's work that he touches on in the journal review article gets at this a little bit. He had a very compelling paper in Nature Medicine 2 years ago with his PhD student at that time, who's now applying for neurosurgical residencies across the country, where they showed that in animal models and in some patients that there was T cell sequestration in the bone marrow, even in the absence of corticosteroids. And so there's something going on here already that's altering the number of the cells there. Now we know that there are some other things that we see systemically. We see increases in immunosuppressive regulatory T cells and immunosuppressive myeloid cells like myeloid-derived suppressor cells and non-classical monocytes and things like this. Dr. Fetchy's work really, I think, was seminal in showing that there's something else going on here in the lymphoid organs peripherally in glioblastoma patients, independent necessarily of the effects of corticosteroids. They were able to track that back to the loss of a protein, S1P1, on the T cell surface of T cells in patients and in mice that had or were bearing tumors of glioblastomas. A recent paper came out from our group here at Mayo. Katya Sufi is a postdoctoral fellow in Aaron Johnson's lab, who's one of our PhD scientists in immunology, that replicated some of this work, but also did some very interesting studies with parabiosis in mice, where the two mice are actually connected, and mouse 1 has a brain tumor, mouse 2 does not, showing that this is very much mediated by soluble factor, that there's the same reduction in, or the same sequestration in T cells into the bone marrow seen in Dr. Fetchy's work. In addition, actually, involution of the thymus. That she was able to track this back down to a soluble factor that was fairly large, so it was less than, or it was the things that were less than 3 kilodaltons, like corticosteroids, had no effect on mediating this systemic response, but things that were greater than 100 kilodaltons were effective. There's been kind of this mysterious glioblastoma-derived soluble factor that's immunosuppressive that people have been describing probably for almost 40 or 50 years now. But this may be another version of that. But very interesting to see this sort of systemic effect on both the bone marrow and the thymus, independent of corticosteroids in animals that had glioblastomas. So that's a paper that was published just the end of last year in Brain. And then sort of another thing that my own group has done working on aspects of this is looking at tumor-derived extracellular vesicles, these little bits of cell that are shed by every nucleated cell. We published a paper last year with Ben Himes, who's a neurosurgery resident in our department and did his PhD in my lab, looking at tumor-derived extracellular vesicles as mediators of induction of immunosuppressive monocytes, myeloid-derived suppressor cells, and glioblastoma, and non-classical monocytes, at least partly induced by expression of the checkpoint inhibitor PD-L1 on the glioblastoma extracellular vesicles. These are potential. They're the right size to be the soluble mediators that we saw in the parabiosis experiments that could be underlying this. But I'll tell you that there probably aren't very many tumor-derived extracellular vesicles floating around in the plasma of glioblastoma patients based on some of our work. But in any event, there's this idea that there's a lot of systemic immunosuppression, a lot of molecular and cellular pathways that we're beginning to understand and dissect that are potential avenues for modulation so that we can get more benefit from the immunotherapies that we do have, whether those are checkpoint inhibitors or vaccines or other strategies.
Well, I mean, certainly not a complicated milieu of events at all, right?
No.
It's this constant interaction between multiple unknown soluble factors and the systemic body as well as the tumor microenvironment. I think it's extremely important work, and I feel like it is something achievable. It's something that, you know, that black box just needs to be open, and we're kind of peeling back the layers every year. The research gets more and more exciting. I think the issue of the journal will be extremely well received. I think, you know, Jason can comment on it as well.
It already has.
I appreciate you guys being here.
I was looking at the journal earlier today, and Ian and Miriam's lead to that has had a tremendous number of downloads and hits already. And I want to underscore just the cast of really experts in the field who contributed to this, including Dr. Gilbert, Dr. Parney, Dr. Fetchy, Dr. Ram, and also an accompanying 4-part webinar series. If you go to the Journal of Neuro-Oncology site, it'll link to the AANS CNS tumor section. There are 4 outstanding webinars that will give you sort of soups to nuts type approach with regard to neuroimmunotherapy. And I think it's a phenomenal free resource that many who are on this webinar will find very, very helpful. We've only scratched the surface in the conversation today, but I was privileged to contribute along with Dan Trifoletti and a few other colleagues on an immunotherapy discussion and review around radiosurgery. And to Randy's comments, it has, or at least it was, commonplace to give steroids at or around the time of fracture radiation therapy and radiosurgery. It's something that we have cut back substantially. I'm much more circumspect about when to give steroids in these patients. I don't know that it's really required, and if anything, in many instances, I'm seeing that the work that Dr. Gilbert showed and others touched upon in this special issue should make us reevaluate some of the underlying assumptions that we've had about steroids and their need around radiation as well.
Absolutely. Thanks, Jason. So guys, we're at the 30-minute mark. Extremely interesting work. Appreciate your time. And this will be recorded and published on YouTube, so you can share with colleagues, and for the viewers at home as well, they can share with colleagues and coworkers and friends. So till next time, I think we're going to be probably taking next week off. We'll be back a week after that. All right.
Thank you, everyone, for
Thanks so
joining.
much. Thank you.
Thanks.
Thanks very much.
Thanks.
Take care, everyone.
Bye-bye.
Thanks a lot, guys. Take care. Bye-bye.